Hello, welcome back to this new lesson of our Virology course. What are we going to talk about today? Well, today we're going to talk about therapy and prevention. You need to know that the most efficient prevention ever is the one you implement in your daily life. Then, of course, there are other kinds of prevention, like the passive and the active one. The passive prevention is the administration of normal standard immunoglobulins, which are obtained by people who naturally faced and overcome many different kinds of diseases, and they were also vaccinated against specific infant rash diseases. The active prevention is essentially the vaccination. Well, there are different kinds of vaccination. There is the vaccination made of killed viruses, the vaccination made of mitigated viruses, that is that the viruses are not totally killed. They still have a pathogenic capacity, a pathogenic ability, but it's just mitigated, as I told you. So it's important not to give these vaccines to immunosuppressed people, for example. Then there are other vaccines made of viral subcomponents such as proteins or nucleic acids or surface antigens and others. Then there are also some vaccines made of viral proteins obtained by DNA recombination technology. So, I want to give you an example of vaccination. So we can talk about the influenza vaccination, because this is the most common one. We have a kind of influenza vaccine made of viral subcomponents. In this case, it's made of the surface antigens such as neuraminidase and hemagglutinin. The advantage of this vaccine is that it can be given to the immunosuppressed people, but it doesn't activate the immune system so much. Another influenza vaccine is made of the viral surface antigens put inside a sort of liposome. The adjuvanted vaccine is a combination of some subcomponent vaccine together with a water emulsion containing squalene, so containing a sort of oil, and a sorbitan triolate in a citrate buffer. So this vaccine is called MF59. And finally, there is the intradermic vaccine. Consider that all the vaccines I told you protect you within starting from 2 weeks after the inoculation, and they normally last for about 6 to 8 months. And what about the passive prophylaxis? Well, the immunoglobulins are glycoproteins, so their life is quite short. They normally die after about 3 weeks. Well, another important thing is that there are some people who really have to be vaccinated and other people who absolutely don't have to be vaccinated. The people who really have to be vaccinated are, for example, the immunosuppressed people, the people who work in public services, the people who are older than 65 years old. The people who don't have to be vaccinated are all the children under 6 years old, the pregnant women, the people who showed an anaphylactic reaction after vaccination, because you need to know that the vaccination is done using the chicken embryos. So there are some people who are allergic to the chicken proteins, for example. Then other people who don't have to be vaccinated are the people who developed a Guillain-Barré syndrome after vaccination, which is a neurological disease. So now we can start talking about drugs. Of course, antiviral drugs exist, but they are really toxic for our organism because viruses are parasites, so they use all cellular structures, and their proteins are also very similar to our proteins. So there are no target molecules we can use to make drugs against viruses. So you need to know that virus infections don't need to be treated with drugs. You have to use drugs only when these infections are really tough, are really dangerous. The drugs we have inhibit the viral cellular cycle at specific levels. For example, at the absorption level, the endocytosis or the transcription of their nucleic acid, or for example the exocytosis. For example, for the influenza infection we have some drugs like amantadine, remantadine that inhibit the M2 protein, which is exposed at the surface of the virus, and this is really important for the fusion of the virus membrane with the cellular membrane. Then there is also a guanosine analog nucleoside called ribavirin, which is always given together with interferon. Amantadine and remantadine have to be given for about 1 week within 48 hours after the start of the symptoms, but the problem is that they normally shorten the disease only of about 1 or 2 days, and they can make our body develop some neurological symptoms, neurological problems, and also the growth of really strong viruses. Other drugs against influenza disease are, for example, zanamivir or oseltamivir, which normally act against the virus antigen called neuraminidase, which is exposed on the surface of the virus. We also have a lot of drugs against herpes infection, like acyclovir, ganciclovir, or cidofovir. For example, acyclovir is a guanosine analog nucleoside, which is recognized by the virus kinase, which monophosphorylates this nucleoside, and the fact that this nucleoside is not a normal nucleoside, it leads to the fact that the transcription immediately stops, and so the viral replication. There are also a lot of drugs against AIDS, like AZT or azidothamidine, which was used in the past for cancer. This is reverse transcriptase inhibitor. There are other drugs against AIDS are, for example, the adenosine or foscarnet. Foscarnet is one of the most toxic ones because this is an pyrophosphate analog, and as you know, pyrophosphate is always used by all kinds of cells for every chemical reaction, so this is a very specific drug. For AIDS, the therapy which is normally used is the HART therapy, which means highly active antiretroviral therapy. This therapy consists on the administration of analog nucleosides, reverse transcriptase inhibitors, and non-analog nucleoside reverse transcriptase inhibitors. The first ones are, for example, AZT or didanosine. The second ones are, for example, nevirapina, efavirenz, and others. And there are also the protease inhibitors such as saquinavir, nalfinavir, indinavir, ritonavir, and others. There is also another therapy used nowadays, which is the CRISPR-Cas9, which is a bacterial system, and this is able to recognize conservative repeated sequences in the genome, in this case viral sequences integrated in our genome. And this is able to recognize them and to cut them off. This system can be also used, for example, to modify the sequence whenever there is a mutation, for example, which causes a specific disease. So this is all for our lesson. I'll see you soon. Bye!